Welcome, everyone. To give folks a minute to arrive. Really excited. I've got some Rhodosodomonas palustris behind me on the screen. Pull it up to the top. But actually, why don't I just switch my screen? I might be able to share it. Let me see if I can share it. No, I'm just going to switch to it. Check this out. I'll drop out probably for a second as it switches. Maybe not. Maybe it was instant. Was that instant? Give me a thumbs up if that was instant. Sweet. All right, guys. So you're on my microscope now. You're inside. You're inside. You see that fuzzy thing? That is a group of bacteria. They're purple non-sulfur bacteria. And you're looking at them. So many people search for these things. And these are the classic shapes right here. And if I backed off the if we backed off on the light more and we went to dark field, they'd be redder. And this sample is actually more brown colored. You can see how it's more brown than red in this. Fascinating. All right. So I'm letting folks in. Folks are arriving. You're looking at a screen, a magical screen. I'll switch my video back and we'll begin. But I just wanted to give you a quick preview of what's to come and what's possible. Am I back? Give me a thumbs up if I'm back. Sweet. Well, I recognize some familiar faces here. Folks are going to continue to arrive, I'm sure. We have over 600 people that RSVP'd for this. People are requesting the ability to record it. It's going to be an exclusive one, folks. Thank you all for being here. This is going to be a lot of fun. I've got a lot to share with you. I've got a framework that we're going to make real in a very clear way today. So I've got some real teaching for you. I want to honor you for being here and taking the time. I'm really excited. There's a lot of folks that just so you know, like in the community already of the Microscope community within my world, there's like over 300 people involved already. So there's a huge movement growing right now. A lot of people are involved. A lot of people are really excited. Many of you are here. I see your names. I know who you are. And that's the amazing thing, is that we really are a community that we know each other. We know the people who are using the microscopes, and the conversations that we have are absolutely next level. And that's one of the ones that we're going to have today. We're going to have a next level conversation together. So thank you for being here. I want to begin because you've made the time to be here. So I want to honor that. All right. If you want, you can click down below and click on the chat and say where you're zooming in from, so you can connect to people that are from all around the world. All right, so we're about to begin. Just pulling up the presentation here and letting more people in. All right. You know what? I think I'm going to find Brian. All right, Brian, I'm making you co-host again, if that's all right, to help let folks in while I'm presenting, because otherwise I'll get all carried up. Indeed, I saw that. You're on. Thank you so much. All right, guys, this is going to be great. This is going to be awesome. More folks will arrive, but I'm just going to begin. I'm so excited to share this with you. Here we go. All right. So let me disambiguate a little bit here. All right. Here we go. All right. Thank you so much for being here. And Brian, thank you so much for facilitating letting folks in while I present. Right. So who's who, the soil biology primer. I'm going to come at it with probably a different angle than you've seen before, especially if you watch the mastermind. We're going to come at it at a different angle. So, but before we get anywhere further, who am I? Let me let in Janet. All right, there we go. All right. So first, but who am I? Well, I am Matt Powers. I used to be a ski racer in New England, then became a musician, play with people like this. I met my wife in the midst of all that madness in life when I was very young. And my wife, when she was very young, got cancer. And it completely changed our lives. It became health became the focus of our lives, and food, and then microbes and all these other things became the focus because it all became tied into how to fight cancer and prevent it from coming back. I left that life. Well, I left the band in New York, and then I went to California and I started songwriting in LA for a little while. And then I did a national tour, but it didn't go that great. And then I ended up subbing in Fresno. Real talk. And so, Fresno is hard. Madera County is really hard. And that's where I became a full-time teacher. And during this time period, I got my master's degree. We had our second son. I started melding cooking and seed saving and heirloom vegetables with teaching. And I fell in love with it. I started doing things that people thought were impossible. This is a corn I adapted to North America from Peru, from the highlands of Peru. No one had created ears, and I used permaculture logic to figure it out. It was permaculture that really unlocked so much of my hope and a lens for solutions and service. So this is a throw-sow garden. I threw the seeds on the ground, and they grew like this. But I'd trained them with several seasons of saving seed from the best plants. So I put this all into a curriculum because I taught at a school that had no books. And so we always were making curriculum. And so I just was like, well, I'll just make a curriculum for permaculture for K through 12. And this was the first time anything had ever been made for K through 12 in permaculture for that demographic, right? And because that was what I was trained for, it was easy and it went through the roof. And this became my life. And so the books went all over the world. They got translated into multiple translations. I worked with people from all over the world. I became a speaker, started helping different organizations, and I became a citizen scientist in the process. My boys grew up. My wife is still here. And health challenges are things that she faces and I face as well. And we try to support each other and do the best we can. But yeah, and then this year I'm really focused on health this year. I know so much more than I ever have. So I'm really, really excited about health. And enthusiasm is, a part of who I am. I love teaching. I love creating curriculum. I've created over 24 books in the past 8 years. So I really focus though on a bridge to a regenerative future. And that's why these products of that focus are kind of like things that are steps on a process to a much bigger goal. So what if you got good at using a microscope? What if you were able to use a microscope to figure things out, to problem solve, to diagnose your plant's health or your compost safety or your soil's fertility? What if that was in a part of your house and you could just go to that room and flip those things on and it's always set up like this? And what if you had that, a home laboratory? And what if you understood your soil and your compost so deeply? Like, I mean, the soil right outside in your garden, but all the soil in your bioregion, all the compost, anything that you you get in contact with it, you understand. So I once read this study that, I've spent a long time thinking about, and it's this study. And if you know this study, you know why I think about it. So it's the exploring the relationship between microscope-based soil biology measurements and tomato yield in South Africa using principal component analysis. So I had heard about the tomatoes, South Africa, the soil lab down there, the Soil Food Web Institute down there, and the success they had. I followed up and found this online and read about it and studied it. And I recognized the methods, of course, but then to see their conclusions and results was worrisome. And there's more, there's more of these types of stories, and we'll get into them as we go. But this was something that was like an alarm bell to me, because as you can read here, it says, when farmers at ZZ2 were questioned about why they were not supporting the soil biology test offered by the Soil Food Web Institute of South Africa laboratory, they complained that the metric was not useful in predicting yield or risk of soil-borne disease. Our results confirmed their observations. So total active fungi and bacteria biomass and above ground pasture productivity, right? What is it they're missing? Fungi, just fungi. Which fungi are they talking about? They have this generalized concept of what fungi is. It worries me. And so I feel like when I read this, I felt strongly that this indicated a need. And when I look at the expression of fungi, whether it's like the fungal hyphae, whether it's embodied in a root propagule, a fruit fragment, top left corner, bottom left corner, yeasts, right? Or bottom right corner, a very large hyphae with crystals on it. So it could be it could be something more like this. And you're like, well, what's that? That's aspergillus. Okay. This is why I'm telling people not to like inhale compost fumes lately, because I'm reading about Aspergillus fumigatus being found in composts, not good composts, but in composts and in garden soils in the UK specifically. So these are pretty identifiable. Okay. But then you're like, what is this? And notice the difference. Dun-dun-dun-dun. Right? You can see it. Everyone can see that there's a difference. Like just hairstyles. Just look, it's different. And this is an IMO collection. This is good. So the context and the subtlety is like really important with this. So which fungi were they looking at? Because in their study, they show no slides, no images. We really can't help them. And of course, there's lookalikes, but they couldn't identify verticillum. You look this up, just a Google search, microscope, you can do 400x, but it actually doesn't really do the same. You could do Google Scholar. It's crazy that the published literature, they favor not showing you clear things. You kind of got to buy books that are references. This is a fabulous book. But it's very clear. This is stained, but it's very clear how to identify these things. So they didn't have access to this information. They were doing those tests that were listed and no others. But mycologists know that you can identify pathogenic and non-pathogenic from their form, or at least down to like tentative, which is actually probably the space that most of us should be expressing a lot of these things that we look at. So my story. My story starts with my family. I'm a family guy. We homeschool, and I have a master's degree in education, and I learned things about the way we do in public school are the opposite of what actually would educate people. And so, um, maybe you were like me and started a garden years ago, and maybe you were like me and you just copied what you saw or read and hope for the best. So I was going off of, Sepp Holzer and just copying, right? And just hope for the best. And they had rationales, but none of it was scientific. You know what I mean? Sepp just lays in fields where they're really sunny and has dreams and then comes up with his ideas that way. That's how he says he gets his best ideas. And I love that. And that's beautiful. And more power to that. I want to do more of that. But we need to know, you know what I mean? Especially when we're communicating with other professionals and who have got like livelihoods and acres and thousands of acres on the line. So I couldn't do that, and realistically and like just hope for the best when it was my family's health on the line. So I started digging and researching. And then it first began, I learned about permaculture and I went crazy. A lot of people go crazy, and like plant everything everywhere and go, bah! And you learn so much in this process because you have like 10% die. You know what I mean? You plant just everything everywhere, and then you have all these incredible things that don't die. And they just become it. There's so much you learn that way. And that's how I learned. I just did things. I experimented, and still to this day, I'm experimenting. Permaculture allowed me to like take, amaranth and just scale it up incredibly fast. How to take landscapes and remediate them incredibly fast. How to grow in 140-degree soils. Yep, that you can see it for yourself. I put it in the soil and saw how hot it was, and then was like, this can't be right, and then started going around and saw that there's this differential, and I was like, and meanwhile it was the same meter and it was 112 degrees outside. So I was like, well, if it's the actual physical thing heating up, all these numbers would change, except they didn't. So the ground was hotter than the ambient temperatures, except where there was mulch. This is permaculture logic, right? This is something we all can do, but it changes how you think and view the world when you're like, oh yeah, it's 140 degrees in the soil right there. Cool. I'm going to grow green plants in that with no water. 3 months in, no water. Like that empowers you in a completely different way. And then when you're like, oh, it drops to below 80 degrees from 140 with mulch and watering, we can do what feels impossible with clear understanding of nature. Permaculture logic is just clear understanding of nature. And that's what I've done and strive to do with all the work I'm doing with soil, is to get out of the way and to create clarity. And so I began to learn about the specific biology at first with EM and then with Elaine. And yes, I know there's multiple factors. There's plant roots. I mean, if you've seen my regenerative soil stuff, it's a holistic thing, but I'm not going to get into all that because we're talking about plant health pyramid stuff here, not the bottom either. We're talking about the top, because only the biology unlocks the top two levels. So you could have everything right, you could be doing everything, and your biology stinks. Well, you're never going to get this top level of plant health. So this is where their immunological health is stimulated to the point of like really pushing things off, really not getting sick, really not getting viruses. Below that, it's like their easy game. But there are higher level predators that, unless you reach this level of biology, they get in. So it's really important that we understand that the biology, the specific biology. I know that John Kemp will go vigorous biology and generalize it, because, you know, he'll sell you the product, right? I love John. I was right earlier earlier this week, but it's specific biology. So the correct minerals unlock the lower two levels, but it's the biology. It's this dance, it's this symphony, it's this communication. But it's while I mapped all of these arrows, right, and made them thin enough to communicate this, all these different layers, the thickness of different avenues is what I would want to communicate, but it would create chaos. It would be just noisy as an image. So we really need to clean it up a little bit in our understanding, and we need to go back to that conversation of which fungi are we talking about, because it makes all the difference. When we understand which fungi and the right microbes that are allowing things to properly cycle, then we unlock the highest levels of expression in our plants. And then we have our soils that are regenerative. They respond to stress with strength and resilience. So in other words, their pH and EH doesn't swing as much. They have buffering from the organic matter, and the structure is electrical too. So it has a buffering on the actual swing of the redox. But it really is managed by the biology. They have the greatest effect. And so we're going to talk about the simplified compost assessment framework today. And we need to know who we're looking at. We need to disambiguate. That's why we're doing this Who's Who soil biology primer. But I wanted to give you a framework so that there's an actual usage for this information for you in your actual practice and life. It's not just, you know, I mean, it's one thing to like just show you what things are, but it's another to give you a tool to actually make a difference in your practice and professionally. Because if you do this professionally and you don't do the things that I'm about to list, you're gonna, woo, because they're easy and they're ridiculously powerful. So I'm really honored that you guys are here today and really excited, because after all these years of study, after all these books, I've got something to show you that is gonna blow people's socks off. That's why, and not only that, it's going to set things up for you to do that for yourself, but also to have realizations that go dun, 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 dun. Because when we understand what's actually at work, we understand the words, and we go through some things, and you're understanding the sentences. You're like, oh, okay, that's a sentence. I can do that. It's a completely different thing when you gain fluency. And that's what my work's all about, is giving you fluency so that you can have a macro to micro understanding, so that you can actually take things into actions and strategies and calendars and you know what I mean? And, you know, recipes and all of it. It's really pragmatic. So I'm really excited about this because these are some of the first like real protocols that like I've drafted completely on my own, and I'm really excited about this because no one showed me any of these things. And yeah, so let's get into it, guys. So step one, do a nitrogen test, okay, on your compost, because every nitrogen test I do on really good compost, and you heard me, really good compost is so high in nitrogen as to be a joke. Do you see that? It's kind of off the charts. It's like neon pink every time. Good compost. Now think about that. What does that mean? Why would that be the case? And the opposite is this. Think about this too. Zero nitrogen in my testing, when it's compost with zero nitrogen, there's no nematodes or protozoa. And if there's any fungi, because there's rarely fungi, it's funky cold Medina scary stuff. So what do you think this means, guys? What do you think this means? So microbial manures have nitrogen. And in fact, if you've been with me for years, it's been embedded in my work. That's the coolest thing about so much of regenerative soil. is it didn't negate all the work I've been doing this whole time. It gives light onto it all in such a new and profound way. So microbial manures have nitrogen. And ladies and gentlemen, you already knew this, right? Because you've been following me. You probably had this on your shelf or the ebook, because the ebook's been free forever too. Hopefully not a pirated copy. I've been giving away free, guys. Look, the nitrate is alkaline. They literally, when you switch the pH level, they change forms. And look at O and look at H. And if you've been with me through regenerative soil, you'll remember oxidized, and one has protons on it. Protons comes from the plant roots, right? So we can imitate all of this and make all of this happen. We can make things more acidic. You can use EM to draw things down. You can use diluted vinegars to draw things down. All of this, all of this. So we can change the expression of things. If you're like, oh, I'm a vegetative state, I'll need the nitrates, but I don't want them strongly. I want to digest them maybe with lab or EM and turn them mostly into amino acids. And when you add them, they're not as a strong nitrate effect. That's why we test. Step 2, pH test. So depending on where we are, there's going to be like a frame of reference, right? pH wise, that you have to be aware of. But if we don't like test the pH of our compost, I've been testing the pH of composts, and oxidized thermophilic composts are oxidized. So they have that O on the nitrogen, right? The O right there, the nitrate, that makes it alkaline. Yeah, pH 8, sometimes pH 9. That's lockout. That's mineral lockout. So we have to keep these things in mind, okay? What we have to do more work if this is your compost, right? Right. We have to take it further, right? We have to take it through an acidification step, a fermentation step. There's options, but it's not going to be beneficial. And maybe you're wetting it and you're getting it to be, because that water is directly related to pH and the hydrogen cycle. It's the power of hydrogen, right? So high pH mineral lockout. So this is why the deserts, hold all those minerals, but they don't give them up until we develop those soils. Step 3, shake and settle test it. You could be doing the jar soil test too, but it really is anything, right? And then you could be like, wait, isn't that all sand on the right? Uh-huh. Yeah, that's bad commercial compost. It's also bad commercial compost. So yeah, no, that's a ripoff. It's a ripoff. And the one on the left is Catalyst Bioamendments. It's absolutely awesome, incredible. Kurt, no drawing, please. um, so I think I can. Well, we'll do it later. Um, the sand on the right there. Do you see how there's that black stuff too? That's not biochar. That's microbial soot. This stuff on the right was taken up to such high a heat that lost all structure, lost all biology, um, and there's a high turbidity, right? Meaning that the water is being cleaned and clarified on the left. Can't do anything. There we go. Let me see if I can clear the annotations. All right, there we go. All right, and we're back. The thing is, all plants contain large amounts of nitrogen. There's a reason why NPK is the primary fertilizer trio that we focus on. They uptake the most of those. And out of all the ions uptaken by roots, 80% of them are nitrogen. Think about that. 80% of what they take up is nitrogen. So obviously, they're going to be nitrogen rich. And this is where all the plant proteins come from. And all organic matter, remember, is nitrogen, oxygen, hydrogen, and carbon. So there would, of course, be those elements in them. And so when we look at compost, we should see that if it was biologically decomposed and it retained the value, which is the nitrogen, the phosphorus, the potassium, all that. If we lose like the organic matter and it becomes microbial dust and sand, and like you just add sand to make up the volume, like it'll look like compost, like that's terrible. Like we have to have the nitrogen tells you that there's organic matter in there that's living. The nitrogen tells you that there is a cycling and release of the nutrients from the lower orders, the bacteria and fungi. That's literally what they tell you. It's soluble nitrogen. So it's really important that we do those steps. People all the time do one thing and they don't develop a holistic perspective. You're like, Matt, I came here to look at microbes. We're talking about all this other stuff. What's going on? But this is what I teach. I teach people to create a holistic understanding and have a 360 perspective, their perspective, so that they develop their eye and understanding and connection to the natural cycles and principles at work. And when we're there in that sensing, observing, and harmonizing, you know what I mean? If we're doing it right, we get the most value and have the most power to respond properly. Not someone 2 counties away or a country away, you. And so that's why it's so incredible to have this holistic perspective and to share this with you, because this is going to change your practice and give you such a deeper understanding of what's actually going on. And you won't get fooled. You won't get tricked like those guys. And there's another example we'll go over soon that's the same deal. So now we're at the microscope, right? And notice I'm showing the epifluorescence in prominent detail because I couldn't do what I do without it. And you can do a lot of the stuff I do with bright field, no doubt. And dark field's very cheap add-on to any bright field. But, and then the manual technique that I do, anyone can do on any microscope. But epifluorescence is so powerful. But we're not here to talk about that today. So do you have fungi? Do you have beneficial nematodes? Do you have beneficial protozoa? These are fundamental questions to be asked after all that work, right? Because you can reasonably guess what's it's going to be like given those tests that you just did. Okay. So here we go. So why is this important? Remember that the nutrition doesn't get released to the plant roots unless it's through, I know like rhizophagy, but it's through the protozoa and nematodes that it is fully cycled and made soluble again. And this promotes the microbes as well, because yes, it reaches the plant roots, but of course it reaches the bacteria and fungi in situ again. And organic nitrogen is fundamental for fungal development. But you don't have a nitrogen source, things will senesce and won't fruit and all sorts of things. So what do they actually look like? That's why we're here, Matt. So bacteria, well, inside, the nodule, the rhizobia, this is rhizobium right here. This is what they actually look like. They're these the clusters of cells. There's different forms that cells take. and it's often stress related to the environment. This is an anaerobic environment. That's why it's definitely okay for them to be forming that form. So sometimes that can be a concern if that's like taking over your field of vision. So this right here, these are lab certified streptomyces samples and images. And you can see that there's spores. This is bacteria. So it's really important that we like unplug our assumptions, because fungal hyphae is what people are saying everywhere online. They're just tossing that around. And if you see hyphae, you might be confused and think it's the wrong kind of thing. And so it's really critically important to understand that like this is bacteria, but this is also bacteria, and they form spores. And then this is this is Azospirillum, and if you think you can see it, you can't. Okay, because that's what it looks like. The little rods, okay, and they're moving really fast, and so they blur out. It looks like, oh, it's those little dots, right? No, no, no. Those little dots are those giant spores, right? So, and then clear fungi, like in this image right here, a lot of people have said is bad. This is Trametes versicolor. Trametes versicolor, turkey tail, primary decomposer all over the globe, is actually a fungal endophyte and is a plant growth promoting endophyte. Let me say that again. In my research, I discovered that Trametes versicolor, which can eat our wood, it has clear hyphae when it's not feeding on substrate that will, because remember, fungi had digestive enzymes externally, so that's how it digests its food. So if you have a pigmented food source, that's why the spores are often colored by their environment and soil type in arbuscular mycorrhizal fungi. So they're clear here. They could be other colors as well, but it's really important to like unplug, step back, look at a larger context, because we want this fungi. We want to partner with this fungi. We want to promote this fungi. But under some frameworks, this has been seen as a bad fungi. So I just want to like unplug all that. This is the typical fungi that people are going to be like, this is great. Clear septa, like a clearly defined dark septa. You've got a darker purplish brown tannish color to it. It's developed. Yes, it seems fatter and thinner in places. It could be damaged by the shaking. But then you also, as you'll read in my book, when you actually examine the morphology of some of the most important species from just a pure culture species perspective, so that you can be sure this is who you're looking at, you find a broad diversity of expression. And it's really important to grasp that. This is probably the most prototypical fungal image that people talk about in soil microscopy. Yet I've never seen it presented this way. This is from my book. As you can see, we go through 40x, 100x, 400x, and 600x, all in the same frame, all the same frame of reference around the single fungal strand. It's really critical to understand that you can see fungal strands, you can see nematodes, you can see all these things, but they haven't been shown like this. So your frame of reference is all screwed up. You're like, well, how big is it really? What is it big in relation to? What magnification is what size? And so I went through and created all that. And then I have little rulers of reference. Um, so you actually can really measure things and understand things. You guys having fun? Let me know in the comments if you guys having fun. All right, these are trichoderma spores. And so it's really incredibly important to understand that like everything has its own unique pathology and tells. And so those spores look awful similar to spores we just looked at. There's a subtle differences, and they're also slightly green if you noticed. And then like EM, these are yeasts. And yes, we're at 1000x, so we're awful close. So just keep that in mind later if we look at EM or something together and you're like, those look tinier than that image. We're at 1000x. We're at the limits of actual resolution. Man, the alignment that day is absolutely incredible. So I'm in the microscope. So what's cool about this image, and I want you to think about, is that epifluorescence lamp is the right side where it shows, hey, some of these are glowing, some of these are not. Well, when you test EM, you'll find that there's no nitrogen and there's no phosphorus. What happens? What do we always say when we put in something with microbes and they digest it? What do we always say? They say they embody it. They lock it up, right? With EM, there's no predators. So the nutrients disappear from the solution and they're consumed. And then we can pull that, and then the phosphorescing ones are they are the ones that ate the phosphorus. So things that autofluoresce at this wavelength of light using this lamp show us phosphorescent material. They show us phosphorus minerals. And in the soil, calcium and phosphorus are mobilized primarily by fungi. And so that's why we can use them in a root situation because the arbuscular mycorrhizal fungi, remember, they're so efficient at it. I would say they're the most efficient at it. Judging, and they're the ones that primarily do this. Because if we look at the saprophytes, they do not fluoresce. Unless they're like Ascomycota, and that's a little bit different. Talking about Basidium mycota, they don't fluoresce. That tells you something about which nutrients they're focused on, right? And what you can transport and then unload. Because yeasts are endophytes. And so you're like, oh, I got no phosphorus in this. But if you're trying to prevent phosphorus from entering the stage of growth that you're at, and you open it up and like you've got all the yeasts are brightly colored, glowing in the middle of the night, that tells you that it's going to deliver a load of phosphorus directly injected in through rhizophagy. So these tools change the game fundamentally. And so I've really tried to open the mind, empower us with tools to look at soil and compost anew and our biofertilizers anew. This is our arbuscular mycorrhizal fungi. Notice how it's different here than the traditional arbuscules, and they're blue and they get the nubby ends. Yeah, that lasts only a little short time and then destroys the sample. The nubbiness and the shape of it is a byproduct of the process, and it's destructive. This is non-destructive. Do you see all those root hairs flinging about? That's because there's no water. There's no cover slip. I tossed that baby under there and just went, boom, we're on. So that there's a reason why it's like that. But that's why. Let me put it back in focus. But it's the same sort of thing. I got to calibrate two things at once. So it's going around the cell walls and then into them. Do you see how some cells are dark, some cells are light? And they're like the square cells you can see, cha-chunk, 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 cha-chunk. Right. It is like that. They're ghost-like. We can see them through the epifluorescence. And then the manual lighting technique. Let me get out of the way with the I can move myself. All right, so That's what a root hair tip really looks like. All right, so let's talk about protozoa. That's what we also that's one of the number one things that you need to be looking for, right? So we're looking at the type of fungi that's there. You obviously don't want to be seeing aspergillus dominating everything, right? You want to see saprophytes in your compost, like the clear hyphae branching. They look like tree roots, right? Well, protozoa, they look like sea creatures. They got these beautiful shells, and they look like sea creatures. And they're hyper diverse. They can have crazy shapes too. These are very very tame shapes here. That's a less than tame shape. Look at that star. It's gorgeous. You can see the bacteria, little dots of bacteria inside it. It's incredible. And those spores are just about the size of its mouth. It might actually be able to take those spores in. What I've discovered in my work, the more you study, the more you see, um, there's a lot of omnivorism focused on the size of the object and the size of the mouth. And that kind of makes sense, right? This is just to show you just the great diversity of like amoebas. There's like naked amoebas. They're crazy blobs essentially with pseudopodia, false feet. But this is primarily what you're going to see, especially in hot compost, Johnson Sioux compost. And they can get very big. Can you see the testate amoeba here? Oh, there's two. It's a bit from my book. You can see the fungal hyphae there on the left, and then the actinobacteria down below. They're significantly different. And then ciliates. Ciliates are often actually trying to help things go from anaerobic to aerobic. So they're actually they are in that transition area when things are waterlogged. They're part of the process that's trying to keep it from going to anaerobic. And then there are, of course, ciliates that are part of anaerobic systems. But it's a wide world out there, and there's a lot of different ciliates. So having tons of ciliates, not good. But one or two ciliates, it's not the end of the world. And depending on the kind of ciliate and depending on the kind of composting you did, it could be you be getting the note from the microbes being like, you need to do a little bit more work, a little bit more time. This pile maybe need to be aerated, maybe need to be inoculated with things, right? um, Biocontrol, um, a new injection of the right kind of life, EM'd, fermented again. Like there's a bunch of things that can happen. So nematodes, these are just tiny worms. They're literally round worms. You're like, wait, round worms? Aren't there those infections? Yes, they are. And they're necessary for the soil, right? So actually, nematodes are the most populous animal or organism on Earth. So when it comes to organisms, there's more nematodes than anything else on Earth. When it comes, like I think there's obviously more bacteria, more fungi, but when it comes to like higher level organisms and animals and worms, nematodes are the pinnacle of population. So can you see the nematode here? We're at 40x. Not so hard, right? You can see the fungal hyphae, right? It's not that hard to see these things. And the nematode's waving at you. It's going, woo, woo, woo. You might see one later today. They're just worms. They hide from the light. They've got different mouth parts that can indicate from this level, but usually you have to go to a deeper level than that, and look at actual examples of who's who, because you can get tripped up and tricked if you just think that root feeders look like the top left and bottom left. Because a Porcelamus, pretty lame, but it's there. And this fungal feeder is an omnivore, most likely. So and most fungal feeders might be omnivores, like a Porcelamus. Diplogastaridae is a switch nematode. It feeds on all these different things because it's got that giant mouth. The mouth size and openness of it really allows it to do a variety of things. This can cut another nematode in half and eat it. Also, look at it in relation to the bacteria present, the other organic matter. This is an omnivore, primarily a bacterial feeder. Beautiful golden lips. This is it in the dark field. But they're just worms. They're clear worms. And so there's an estimated, they don't know, there's an estimated 2 to 3 million species of nematodes. That's their greatest educated guess. They have no idea. And there's tons of lookalikes. Most, from what I'm seeing in my experience and all the study, is most of them will eat whatever they can get their mouths around. And their mouths, that's why we look to their mouths to determine what they're most likely to eat and prey on and be good at preying on, right? They may like try to feed on things, they be messing with things, and you're like, looks like it's kind of messing with that, but it's not got the mouth type to do it. It's not going to do much damage, you know? But these worms are, survivors, and they're going to do what they can. So let's look, right? So this was, let me just, we're going to pause this. We're back. Thank you all for being here. And we're going to switch to this. So we were just looking at this, which is a culture. We were just looking at, we were just looking at this, which is a culture of purple non-sulfur bacteria. You can get it at Algae Barn. So Rhodocytomonas palustris, incredible culture. Important to have frames of reference so you can compare things and know what things look like in different conditions. I'm gonna let this settle for a second, but this is Catalyst Bioamendments compost diluted 1 to 10. So build it up to 1 with soil, well, with compost, and up to 10 with liquid, and then it was already shaken. I'm just getting it to be. The thing is, you have things settle at the bottom like nematodes, and so I want it redistributed. So when I take something out of here, we're more likely to have a nematode in there. Usually, there's a nematode to up to 3 nematodes. Usually, like 1 to 3 in that range in each drop. They've got such good compost. You don't want to take it from the top. You don't want to take it from the bottom. And you also don't want to take it and then pull it out. You want to, so much of what we do is circulating super gently, especially when we get to the micropipette level and start doing DNA together. Yes, very, very carefully mix the 2 fractions of a drop together. That'll be what happens. So I'm going down, not all the way, and then I'm pushing back and then bringing it back again and mixing it. But I don't want to disturb that lower layer too much. So typically you're going to want to like get in focus. I was already in focus, so I should be able to just go right up there. Don't necessarily do that on your own. I know you're breaking things. I just knew where I was, and I do this like every day for hours, so there's reasons. I can do that, and you'll learn that too eventually. So already at 400x, I can see that we have an average of 1 or less than 1 actinobacteria per field of view. And you can diagnose things very quickly just by doing fields of view, random fields of view like that. Spores, actinobacteria, lactobacillus, things that are of lower counts usually. I've got 2 lactobacillus. I've got 3 actinobacteria and 2 lactobacillus in this image. You know what? I forgot to share the screen with you. I'm just realizing that. Let me share the screen with you. Haha. All right. Get all caught up. That's the thing is I get like sucked in. All right, ladies and gentlemen. So lactobacilli, you can see the bottom left. There's a strand of that. And you want to travel down and see, you see that in the center there? That really long, you know what? I might need to do it over here. So I'm traveling through the layers. The lower part and the upper part have different things. And I am also, I have different screens that have different brightness. So I'm going to try to use the screen you're on to find the thing. Wow, I'm so blown out over here. Weird. Oh, it moved off screen. So part of this is when you put the drop first on, you put the thing down like that, everything moves. And so letting it settle for a minute really helps. But let's find a nematode. So let's go up to 40x. So this is typically what you're going to do. You're going to start off here. Start off with the lowest light. And you're going to move it up slightly. You're going to get yourself in focus. I'm just looking at it here just to make sure. All right, so now here at 40x, let's move around and find some nematodes. Do, 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 do. So it's a little like Where's Waldo? Glad you're seeing the screen and not the back of my head. This is working so well. Don't you guys think this? There's a nematode. I told you all. We are there. Do you see the nematode? Raise your hand if you see the nematode. Here, I'm going to try to sharpen it on your end. All right, so here we go. What we're going to do now is we're going to start jumping in. And it's wiggling, so at a certain point, it's going to be like, what are you doing? And it's going to be hard to track, but let's do it. So what happens when you first zoom in at 100x, you're going to like you see how it's forming a loop? You're going to go inward with your focus, your fine focus, into it. And then when you go up to 400x, you're going to go back. So it's a little bit rocking back and forth. So beautiful specimen. Let me turn up your light. The thing is, I've got to turn my light down So what I use is a brightness compensator on the actual, oh, you can see that, can't you? Yeah, you can see that. You guys, do you see the brightness compensator? So hold on. Let's see if it affects you. Oh, it does affect you. It's just, I need to be like blown out on mine for you to see it. Okay, this is so sweet. So I'm going to move this down and you see how it reacts backwards. Did you guys see that? That's a critical tool. So I can go extremely low light on this and then he calms down or she calms down, you know? Here, it's completely off now. And we go up slightly. There, I've gone up just enough. And it obviously doesn't like the light. That's why it's freaking out. Let's get a little closer. So we're here at 100x. And then you kind of have to find your way again when you go up to 400x. That's 4 times. Oh, look at that. So what do we know what that is now, don't we? It seems to have bent your image a little bit. Does it seem like that's the case? It's bent the image slightly? Yeah, it's stretched it on my end at least. Does it seem like that? I wonder if I can fix that. Interesting. So that's a testate amoeba right there. Yeah, it looks bent. Or maybe it's my screen on this computer. Huh. I don't know. All right. So that looks a little bit better. So that's testate amoeba. Let's find that worm. Hey, buddy. Now, do you all see the utility in being able to film and take an image and then look at it afterwards, slow down video, or to see what that still image looks like? We can study its behavior. And I know there are people who recommend like flaming the slide and all this stuff. They give up eventually while you're doing your other tasks when you're evaluating soil or compost. Yeah, that's a bacterial feeder or a bacterial feeder omnivore. Let's see if I can get it to be brighter for you guys. There we go. So you have to kind of chase them around. And when you're videoing, you can capture all of this and slow it down, and then you can much more easily identify what their mouth parts look like. So this is so cool that we can just share this image. I haven't seen anyone be able to share their microscope over Zoom. So I was making sure that I could do this earlier today. I found out like 3 ways that I can do it. You can connect like photo booth. There's like all these different things you can do. So this isn't even the device I'm using is designed for a PC. It's not even designed for a Mac. So the one thing I'm certain, it's not a root feeder, right? You guys didn't see the telltale barb and the bulbs at the end, and it didn't have the lips, the shape. It had the concavity in there. That's like that's like a trough in the mouth. So we know there's a few things it could be, but we know what it isn't, right? And that's more functional in this scenario. Like I said, we're just doing compost assessment. simplified here. and and that's the beauty of it, is you actually don't need to like break out the, because I have the reference textbooks, right? I can go through and we can start identifying the fungal types, and you know what I mean, all those kinds of things. But I already knew the type, and that's what mattered. It was Basidium mycota or Ascomycota, you know what I mean, or Zygomycota. And you're like, well, why don't we just see what it's been eating? Ah, so do you see it's not eating fungi? Remember that image I showed you where the stomach contents were glowing? Well, this one, it's not. More evidence that supports that it's a bacterial feeder. And we saw its behavior. You know what I mean? It's going out and capturing little bits all around. Those little bits are bacteria. And they're obviously at a different frame of view, and that's why we see these big halos around their shapes rather than them. Bring it down to them. They're more like this. So so the key is playback, you guys, when it comes to so much of this, because it's like someone's asking how you get a sharper image. Um, you catch the moment when it comes to this. You either kill it, wait, or you catch the moment. And there's moments like, watch this. Like all here, these are, if this is videotape, all these moments are in focus. So so that's what I'm trying to say though, is that if you don't have a good camera attached to your microscope, identifying who a nematode is becomes much harder. And I know you can like flame it and slow it down, but even then you're going to want to be able to understand the complexity of the situation with, and have a reference to look at the mouth parts. And you don't need to even do that because we just saw it enough that we know it's not a root feeder. We know it's an omnivore or a bacterial feeder, and that's good enough for our purposes because we know um, we can go deeper. But for today, with a simplified compost assessment, that gives you a real clear benchmark of where you're at, what's cycling. You've got protozoa and you've got nematodes. That's the number one thing that everyone I talk to who does compost assessment never sees. And that's the root of the issue, is everyone's stuff is just the actinobacteria. It's let me see, let me just get you guys in focus on this end. So this, don't see, oh yeah, I do. There's septa there. Can you guys see that? Right there, at the right above where the turn is. So that's fungi. It's Basidium mycota, most likely. Man, it is hard for me to I might lower your brightness just for a second so I can see it on here because it's much easier to see than on that screen. Let me let this person in and then let me show you a couple more things. Yes. Okay, so this is a great example. This right here. It's fascinating because what you guys see is a little bit different than what I see, um, just like brightness wise and clarity of the image. All right, there we go. Do you guys see how that's thin and clear? This is actinobacteria. Um, and so if we turn this off and we look at this, you can see a fungal activity at the base. See that green light there? So this is really not as fungal, the organic matter here. But that makes sense. Because that's actinobacteria. That's bacteria, not fungal, right? So the activity there is not being controlled by fungi, but actinobacteria in the specific organic matter particle. So we can see fungi and we can actually jump back out. It's easier to jump back out here to 100x to find fungi. We could see fungi at 40x when it's giant, but when it's at 40x, you can really scoot around and see a lot without having to be so close, like having it close to your nose. Ooh, look at this one. Oh my gosh. This is going to be awesome. So I'm going to turn up the light again for you. And the brightness, actually, specifically. Okay. So they look like little roots, right? You see the little leg winking? One's dark and one's light. It could be at different distances. It could be different fungi even. Let's look. It's so bright. I gotta back it off to actually see what's going on. So there's clear septa. And this could be a few different things. It could be Basidiomycota. It could be young Trametes versicolor. It's a bunch of things that it could be, but it's definitely fungal. It definitely has clear septa. It's weird that it's a distance. That's the thing, is it's only showing here. Let me get that out of the way. The quality of the image is definitely not 4K that's being communicated over the screen, unfortunately. Just want to like put that out there. It's a much bigger image, and they slightly resize it, which is frustrating. But I really appreciate you guys being here and checking all this out with me. We just looked at Catalyst Bioamendments Compost. I'm going to hop back out here and continue the presentation because there's more to share. Was that cool? Was that cool? Did you guys learn something? Let me know in the comments. Give me a thumbs up. Let me know. All right. Oh, thank you so much. Thank you for being here. All right, so we did look and we learned I want to tell you about a tale of two labs. So they had the same sample given to them by a savvy farmer, and they were both certified soil labs, and they gave different recommendations. And then the farmer revealed it to the public. Oof, right? Undermining, right, is rough. And it's because their protocols gave them different numbers. And this was entirely preventable if they had done this in front of each other before and compared their notes. They'd have been like, wow, none of these things add up. That's why people repetition, trying things different ways, stretching things, doing things. This was preventable. So if we want better soil, we have to become citizen scientists. We actually have to look ourselves, test ourselves, because current tests and methods are just formalized experiments. And science is always in a process of developing new experiments for deeper insights and from deeper insights. So current test methods were always formalized experiments. And if we want deeper insights and we want better soil, we can't follow an old formula and expect to get new insights. We must create new insights. We must go further. We must take the time to look, because if we don't look, we don't see. This whole webinar is about showing you the unseen world and how connected it is to the world that we're living in now, how dependent we are, and how you can understand and further the understanding for everyone in the community and in the world, because it is cutting edge right now. All the paradigms are shifting. All that was old is being thrown out. All that is new is just being established. Our understanding, our paradigms around soil, around compost, around plant health, around health in general, all of it is changing. We need new tests. We need new insights. We need to ask new questions. So thank you for being here. If you want to learn more, please go to regenerativesoilscience.com. I've got books and reviews and courses and more free things. So check that out. And please share when the replay comes up. Please share the links to these things, the sign ups. Please share, because this information I've never seen anywhere else. This information will transform your understanding and other people's understanding of the world they live in, of their soil, of their compost, of the food they grow. And that will empower them to make better decisions, to make better food, to live healthier lives, and to help their community at a higher level. If you want to learn more, join us in the next webinar in this series, and join us for the live Q&A, which is happening right now. Thank you for being here and taking the time to be with us. All right. Wasn't much more, but I wanted to end with that thought, so that really framed our understanding. We need to be asking which fungi. We need to be looking at things holistically with our thinking caps on. And when we do, amazing things will open up for us. That's what I've experienced with writing Regenerative Soil Microscopy. Completely new insights. And I'm so excited to share them all with you. You know, it's so wonderful. So many of the people answering the questions and diving in and helping me, because I see so many of the questions. There's like, oh, those are my students that are answering those questions. Like Brian Crump, Crystal, Bianca, Laurel, Patricia, Scott. These are my students. So it's like the teacher's joy to see that, because it's like, you guys, you get it. You are the citizen scientists. You're out there, you're making it happen, and you know it. And it just comes readily to you, because it's not me. This is nature we're talking about. And so you can go out and verify it. You can go out and test it. You can go out and observe it and experience it. You can go and grow it. So it is an honor to be your teacher. It is an honor to train you today on all this stuff that I've never, shared this stuff before. I've never gotten to share the screen, the microscope with you and talk at such a deep level with you guys. I can't wait for next week. We're going to keep diving into the Discover Soil Microscopy webinar series. I am just over the moon. I'm so excited. And I hope that you have a wonderful weekend. I'll put a replay of this up and I'll send you guys an email about it. I'm Matt Powers. Thank you for being here. Grow abundantly, learn daily, and live regeneratively. And I'll see you guys soon. All right. Have a wonderful night.